of the meeting that we're having today. We are encouraging that you utilize the chat box to ask questions as the presentation will be going on. You will have a chance to be able to mute yourself and ask your questions at the end of the presentation. During the course of the presentation, though, we ask that you remain muted at all times. We will also share in the chat box a link, and we're asking that you complete that evaluation form, which will help keep improving this series. So for now, I would like to pass it over to my co-moderator, who will first of all introduce himself, then introduce the speaker who he will call to give the presentation. Amos Evans.
Thank you very much, Prof. Dr. Catherine. So I also put on my video briefly. So my name is Evans Xose-Amuzu. So I am a public health research officer, and I currently manage sickle cell clinical research studies at the Komfo-Anokye Teaching Hospital in Ghana, and also the Ghana site coordinator of SPARCo, which is part of Sickle in Africa. Those are my main duties. Just a little about myself, then I'll move on to our speaker, our distinguished speaker. So Prof. Hamosh is the Dr. Frank V. Sutherland Professor of Genetic Medicine at the Johns Hopkins University School of Medicine and the Scientific Director of the Online Mendelian Genetics in Man, OMIM, since 2002. She served also as the Clinical Director of the McKusick-Naton's Department of Genetic Medicine from 2002 to 2025. Dr. Hamosh received a BA in Biology from the Wesleyan University, MD from the Georgetown University, and MPH from the Johns Hopkins School of Hygiene and Public Health. She also completed a pediatric residency and clinical biochemical genetics fellowships at the Johns Hopkins University Hospital. Then Dr. Hamosh has also authored over 140 papers and serves on several international committees presenting genome and phenome relationship as well as phenotype ontologies, including the CleanGene Project, the Global Alliance of Genetic Health, and the Human Genome Organization, HUGO. She served as a two-time president of HUGO, that's from 2023 to 2025. Then she and her colleagues also developed the PhenomeDB, which is a web-based tool for the collection and storage and analysis of standardized phenotypes and genotype data, and it's freely available to all. And also the Gene Matcher, which is a website which also matches clinicians and researchers with interest on the same gene. Then with that Gene Matcher, there have been over 17,000 submitters from 114 countries with over 100,000 cases. And matches in these have created over 1,000 publications, which have described over 700 novel disease genes. And Gene Matcher is a founding member of the Matchmaker Exchange, MME, and Dr. Hamosh has served on the steering committee of the MME and represents it on the steering committee of the Global Alliance for Genomics and Health. Quite a very elaborate experience for our speaker. So without much ado, I want to invite Prof. Ada Hamosh to speak to us on OMIM and its other useful genetic resources for Africa. Over to you, Prof.
Great. Thank you very much for the kind and long introduction. I'm tired already, and I hope you guys from hearing all about myself. And actually, this is the title. I should have changed the title. Today's title is going to be more about how to use OMIM to find out about genes and biology, because I understand that this audience is a mixture of master's students, graduate students, some postdocs, and some MDs. So I'm going to, oh my goodness, I'm going to turn that off. Sorry. And then I am going to, sorry about that. And then I'm going to just tell you about how you can use OMIM sort of as a portal to find out more about genes and disorders and their relationships. So, okay, here we go. I have a tendency to get excited when I talk and speak even faster. I'm going to do my very best not to do that, but if I do it, just somebody tell me slow down, and I will. So, in the beginning, there was Gregor Mendel, who found out all about inheritance in peas. And then there was Victor McCusick, who studied inheritance in humans. And this is his official portrait at Johns Hopkins. I very much like this portrait because he is sitting in his favorite chair. Behind him are the many editions of Mendelian Inheritance in Man. On the left and on the right are 3 editions of Heritable Disorders of Connective Tissue. Both of them are books that he authored and went through many editions with. And directly behind him is a photograph of the Amish Madonna. He did a great deal of his work working among the Amish. And he and 2 other men, James Neal from University of Michigan and Arno Metelsky from University of Washington, are considered the fathers of genetics. In 1957, all 3 of them independently started genetics clinics in their various universities and hospitals. Okay, so Dr. McCusick liked to take notes. He started out as an internist, then a cardiologist, and then went and created the fields of genetics, and everybody thought he was ruining his career. He did okay. So, which means follow your heart, by the way. So, these books were called Mendelian Inheritance in Man, and initially they were titled A Catalog of Autosomal Dominant, Autosomal Recessive, and X-linked Disorders. But around this, can you follow? Can you see my mouse? The title became A Catalog of Human Genes and Genetic Disorders. The first edition had 1,600 entries, and the last edition in 3 volumes had 8,000 entries, and it was printed on like onion skin, really thin paper. And now we have almost 28,000 entries, so we can't publish this as a book. So MIM was always prepared for computers, and in the 1960s, computers, the cards, the photography for the cards, everything was in capital letters. And so MIM entries still have all caps, and because it was a book, it's organized as if a book has an index, right? So you would not want 500 idiopathic, right? You'd want pulmonary hyperplasia, comma, idiopathic. So that's why it's organized that way. And it stays as capital letters, which I actually really like, because if I'm trying to figure out where somebody found something, if it's all in caps, I know where it came from. It came from my resource. So in any case, always computer ready. And it's always been part of actually using information retrieval and natural language processing before anyone was thinking about AI and large language models. It was one of the very first resources to be available online, which is why it's called Online Mendelian Inheritance in Man, which seems like a funny name in 2025, but it was really quite revolutionary in 1987. And it was hosted at the Welch Medical Library at Johns Hopkins, and paid for the servers in those days, in 1987 dollars, was $20,000. Now that server would cost you maybe $1,000, probably less. But in any case, it was available by dial-up until 1995, when it was integrated into the World Wide Web by the National, oh my gosh, it's the Information Biology Institute, National Center for Biological Information, that's biological informatics, at NIH, National Institutes of Health, and it was incorporated into their Entree suite and looked like that. And it stayed there until January 2011, when we developed omim.org, which is a resource that we hold with extensive links to everything, we being Johns Hopkins, with extensive links to everything else. So this is the history in terms of the growth of entries in OMIM. And we're almost, by next month, we'll be at 28,000. So it continues to grow. This is the information, and by the way, omim.org is where you go. It's a very simple URL to remember. So this is the information framework. So OMIM contains information about phenotypes and about genes. Phenotypes we define as it's basically a disease, a disorder, or a recognizable constellation of features. So some of them aren't diseases. It can be sort of a combination of, it can be, for instance, the ability to curl your tongue. I will show you myself curling my tongue. That is an autosomal recessive trait, and it's in OMIM. So there's all kinds of things. Phenotypic entries have a clinical synopsis, which is a regular feature or list of the features of a condition. I'll show you those in a few minutes. And then every entry has specific topical links from that entry out to other resources. The link only exists if there is a link, a page in the other resource. If there's nothing about that condition or that gene, the link won't exist on that page. Then there are gene entries. Gene entries are where variants are found, but not every gene has variants listed in OMIM. I'm going to talk to you more about that in a moment as well. And they have different external links, the kind of things you'd expect about from a gene. And those links, again, only exist if there's a correct page on the other resource. So you go right to the page you want. The relationship between phenotypes and genes is created in the gene map. And from the gene map, we create, and everything is done by hand by humans who are experts. None of this is done by computers. A phenotypic series, and we also create molecular series, and those can be shown in different ways visually and gene graphics. So I'm going to go through all of this material in the next little bit as fast as I can without making it crazy. And every gene or phenotype and entry is assigned a unique 6-digit MIM number that is stable no matter what happens to the name of either the gene or the disease. Okay, so that's what the first part is. And these MIM numbers, that's what they're called, MIM numbers, are used extensively in the literature. Then there are the main title, the current used titles, and then many other aliases and symbols, including ones that are no longer in use. They'll say comma formerly. But because this is next year will be 60 years old. OMIM has been around since 1966. If you're looking at older literature, you might not know that you want certainty you're looking at the same thing. Is this this disease that we call this now the same as what we called it then? Yeah, it'll be there. The aliases and symbols will be there. There are rich full text descriptions, but they're very structured in the way that they're presented, and so it's easy to compute on OMIM. And everything in OMIM is cited to a paper, to the peer-reviewed biomedical literature, occasionally to a book chapter, not often, but occasionally. And those are all those citations are available within OMIM. Okay, this is the little tiny group of people that work on it. And these are all the people that have contributed to MIM and OMIM over the years. This Kelly Preslepa continues to work. Everybody else is passed. No longer working with OMIM. They may be alive and well. Sorry, let me be clear. Okay. So this is the growth of gene phenotype relationships in OMIM. So in orangish are the number of genes with a variant that causes a phenotype. And this is not a whole year, which is why it looks like it's slowing so much. It's not, it's just not a whole year. And then these are the number of phenotypes with a known molecular basis. And what you can see immediately from the graph are two things. One, it's going up, still going up. And two, there are always more phenotypes with no molecular basis than there are genes, which implies, and it'll be a theme, I'll be talking about this repeatedly, that there are often 30% of the time when there's a known disease gene, that gene is associated with more than one disease. And sometimes they're a little bit overlapping, not clear it's really two things. Very often they're completely distinct disorders, either by inheritance pattern or by the effect of the mutation of the protein domain. So we'll talk more about that in a little bit. Okay, so this is the homepage, omim.org. That's where you go. This is the search box. It's very simple. I work with old school programmers and they want it to look clean. So you won't see any flashes or videos on the home screen. So we describe over 17,600 entries as genes. We define a gene as a functional element of the genome. So it could be a protein coding gene, it could be a microRNA, it could be long non-coding RNA, it could be a locus control gene where a region, not a gene, doesn't code for anything, but where it's very important to the effect of the other genes around it and more. So if something, and even enhancers, the breakpoints of TAD domains, which are little locus control areas, all of those things could be described as a gene in OMIM. We have very, very broad definition, so we can include everything. More than 5,000 entries have variants, and there's close to 36,000 variants in OMIM. But 36,000 is actually a very small number because in OMIM we only do representative variants, and I'll tell you more about that in a minute. So this is an example of a gene entry, and I'm going to just give you the anatomy of a gene entry. So in this case, I searched for fibrillin, and at the top here, this is the MIM number, that 6-digit number that describes fibrillin 1. This little asterisk means that this entry describes a gene. Okay, and I'll show you more about that in a second. Fibrillin's a little bit boring. It only has one alternative title. Almost always, OMIM uses the official gene symbol as the gene symbol of our entry. On rare occasions, we don't, but this is always present right there, and that's a live link to the Human Gene Nomenclature Committee approved gene symbol that takes you to that page of that resource. This gives you a set of genetic location and the genomic coordinates. If any gene is related to phenotypes, has associated phenotypes, this table will exist. This gives you the list of what they are. You can see these are all autosomal dominant variants. And if I wanted to see what they are, because I don't know what these are, I can click this button, which I'm going to do right now. And what that shows me something called side-by-side clinical synopses. So these are the 8 things that are caused by variants in FBN1. And what you can see, even if you've never heard of any of these things and have no idea what I'm talking about, is that some of these you're short, tall, short. Can't be the same thing because some you're tall and some you're short. And you can go along and you can see that there are additional differences. So these are very different disorders. Going back to my anatomy lesson. So this side of the table has the table of contents for the MIM entry. And if you click on any of the things, you go to that section. We're going to go in a minute to the table view. This section over here has the external links that will go to other resources, to the FBN1 page of other resources. Okay, and then these are gene graphics. We'll get to that in a minute. Okay, so I've now gone to the table view of the allelic variants, and I see that there's 70 selected examples. If I click on this, I can find out what kind of examples, and I'll tell you that in a minute. But this will take me to all ClinVar variants. So the OMIM has 70. What's going on in ClinVar? And I'll tell you more about ClinVar in a minute. So I'm going to go to ClinVar, and I can see that, oh my goodness, there's almost 4,000 different pathogenic and likely pathogenic variants in FBN1. So you can see that OMIM gives you example variants, but if you want to look up a variant or know more about variants, you go to ClinVar to find out what's going on. Okay, so ClinVar is a database of reports of the relationship among human variants and phenotypes with supporting evidence and history of the interpretations. It is an aggregating resources. It's giving you what other people have said about different variants, and I'll show you in a moment. And the interpretation of genetic variants changes over time, hopefully not the benign and the pathogenic, or even the likely benign and likely pathogenic, but there are very many variants of uncertain significance, and those you have to follow over time because the interpretation will change as more evidence accumulates. So you go to ClinVar to look up the genetic variants. And again, I always go to OMIM to get to these other resources because I find it easier. Okay, so I've now gone to an FBN1 specific variant. I can see up here that this is a germline variant, and this classification is 2 stars. So the stars are not about how bad it is, it's about how much evidence there is. So no stars, if it were only an OMIM, it would have no stars because that's based on the literature, and we don't interpret it according to the full standards of what ClinVar wants. We're just putting, our emphasis is putting in new gene phenotype relationships. If it has 2, that means there's more than one submission. One means one lab has submitted, or some contributor that's a bona fide contributor has submitted a variant. That's 1 star. 2 stars means more than one, and they agree. You get no stars if there can be multiple submitters, but they have conflicting interpretations. 3 stars means an expert committee has reviewed it, and 4 stars means it's part of a guideline. Okay, so that's what those mean. It's the strength of the evidence. It could be for being benign. Okay, it doesn't have to be for being pathogenic. It's just the strength of the evidence. Okay, moving further down on a page, you can see this is the name of this variant. It has a variation ID. It has other IDs. And then it was first submitted in April of 2018. The last time this specific variant was submitted by a lab, any lab, was in April. And the last time it was formally evaluated, where somebody dated a big evaluation of that variant, was in 2021. Here's all the different HGVS. Sorry, let me go back one for a second. HGVS stands for the Human Gene Variation Society, and they make the rules about what to call a disease. I mean, sorry, wrong, totally wrong. What to call a variant and how to put it on the genome. And they want you to specify the transcript. And in general, everybody's using something called the main select transcript, which is the longest transcript. And it's something that both the NCBI's RefSeq and EBI's Ensemble have agreed on. This is the transcript that we're calling. This is in, where is my mouse? There it is, sorry. This is the, in the RNA. This is in the protein coding sequence, or this is cDNA really. And it tells you what the consequence is of that variant. Okay, then if I scroll further down for this particular variant, these are what people have said, and this is the date that they submitted it, okay? And this is who said it, and whether they put in review assertions, what they said it caused, okay? So these are different calls, who they are, and then you can read more about what they said, okay? And then I'm going back to my anatomy lesson. The next thing I'm going to show you is clean gene graphics. This little question mark takes you to a PDF that explains what's in here, which I'm not going to tell you now because you can read it yourself later. And then I've done this for FBN1. So this view is called the linear view. No hierarchy is implied, but here's FBN1. Here are its 8 phenotypes. And you can see here that geliophysic dysplasia is part of a phenotypic series for geliophysic dysplasia that has 2 other genes, ADAMTSL2 and LTBP3. While Marcasani, also caused by FBN1, is part of a phenotypic series with 3 other genes involved, and so on. In this particular one, this is a Marfan lipodystrophy progeroid syndrome caused by FBN1. It's part of a progeria phenotypic series, etc. So this just lets you learn more, and you can pick another one of these and it'll rebuild this for you in a different way on the fly in real time. So what you see today, if we change it tomorrow, might look different. Okay. This is called the radial view. Here it puts FBN1 in the middle, and this is the progeria phenotypic series. This is the geliophysic. This is the wild Marcasani. These are the other conditions that are part of a series because there aren't other genes involved. And you can rotate this. Right now, FBN1 is in the middle. You can pick that, etc., and you rotate the figure. So it just depends on whether you like to look across or rotate. Now, I've now gone into Marfan syndrome. So I'm going to give you the anatomy of a phenotype entry. Okay, so in this case, this is preceded by a number sign or a pound sign or a hashtag or whatever you like to call this little symbol. In OMIM, that means that this entry describes a phenotype whose molecular basis is known. Okay, so that's the MIM number. This is the name. Also a little bit boring, doesn't have a lot of other names. There is a phenotype gene relationship because Marfan syndrome is caused by mutations in FBN1. These are all live links. Okay. The other thing I want to show you here is there's this little thing, it lives up here, it says ICD+. If you mouse over it, you open this up, and you can see other codes, SNOMED CT, ICD-10, Orphanet, the Orpha code, disease ontology, and the Mondo disease ontology, monarch disease ontology, Mondo. They're codes for this disease. Okay, so these are synonym codes from other resources. Okay. We're going to go to the clinical synopsis in a minute, but I just want to show you that this is the table of contents for the phenotype entry, which is different. Okay, there's standard headings here. They're not all used if we don't write something about them. And again, you can see external links, and this one opens to clinical resources. The other one opened to variation and genes, but all of these are toggles that you can open. Okay, next, I'm going to show you the clinical synopsis, I'm pretty sure. So this is a clinical synopsis for Marfan syndrome. Everyone begins with inheritance. It's got a standardized mechanism of of being presented. The bold and the italicized words are stable. They do not change no matter what. The other words may vary. They will vary from entry to entry, but they're a semi-controlled vocabulary. It's not exactly human phenotype ontology terms, but I'm going to show you more about that in one moment. So in these little busts, see these little busts? That means this word, you can mouse over it to see a picture of it, and you can click on this to get a definition. So if any of you are unfamiliar with these very unusual dysmorphology terms, it's a great way to figure out what they mean. So in this case, this is a baby with dolichocephaly. Dolichocephaly is a long head. And if you click on elements of morphology, it will take you to elements of morphology. And here you have the definition. You have the same pictures. We took the pictures from there and the comment, etc. Okay. So you can do that for anything that has a bust will take you to that. The other thing I wanted to show you, this is the same information. I've now clicked a thing called feature IDs. And here, if we go through the clinical synopsis, and don't do this, just as a way of telling you this is all coded in the back end, are the SNOMED CT, the UMLS is Unified Medical Language System, HPO is Human Phenotype Ontology, and there are some others in here. These are all available. We mine them computationally, and you can also go across OMIM through an API, which I'm going to show you in a few minutes, that allows you to see all this. But of course, don't do this piece by piece. You will lose your mind. This is for computers. Okay, so the clinical synopses are available in a variety of different views. I showed you like just clicking into the clinical synopsis from the entry. The other thing you can do, in this case, I have searched progeria, okay, and I've gotten this retrieval. And this retrieval, that's a phenotype, that's a gene. That's a phenotype, that's a gene. That's a phenotype. So if I only want to see the entries that have clinical synopses, I click this button. This is clinical synopses, and that gives me this view. So it got rid of the genes immediately because it's only things with the clinical synopses. And then it has these highlighted section headings, body parts, that systems, that word is systems, that if I mouse over them, I can see what's in it. So that's one way to look at it. The other way to look at it is what you just saw. I can pick these two and ask to compare them, and I get the side-by-side clinical synopsis. So it just depends on how you look at information. It's all the same information. It's just different presentations. Okay. So what kind of phenotypes do we include in OMIM? Well, obviously, for a database called Mendelian Inheritance in Man, we talk about single gene Mendelian disorders, obviously. Cystic fibrosis, sickle cell disease, achondroplasia, many, many, many others. We also talk about phenotypic traits that are not necessarily disease or disorder. We do talk about susceptibility to drug reactions, malignant hyperthermia, warfarin sensitivity, many others. Altered susceptibility or reaction to infections, including herpes simplex encephalitis. These are examples. Herpes simplex encephalitis, progression of HIV to AIDS, severity or protection from severity of different kinds of malaria, so falciparum, etc. Those exist in OMIM. Germline susceptibilities to cancer are in OMIM, BRCA1, BRCA2, many others, but we are not the place to go for somatic variants or variants in genes that cause somatic cancer. These are really germline susceptibilities that we focus on. And then we also report recurrent deletion and duplication syndromes that are relatively common. We are not the resource to go for unique, never reported variants or reported only once or twice. There is another resource actually called Unique that goes over, it has wonderful information for not only clinicians, but patients and families about those. Okay, so I don't know how many of you are clinicians, but it can be very challenging to define a phenotype. And these are some of the challenges. There are changes in diagnostic modalities, so you might not have been able to see that something was a feature until something new came up. So my usual example for that is a condition called Loeys-Dietz syndrome, in which you have remarkably curly vessels. It's called arterial tortuosity. And you could maybe have surgery because of a problem and say, oh my goodness, those arteries look funny, but you'd think that's just that place. But now we have the ability to look head to toe at arteries using something either called an MR angiogram or a CT angiogram, and you can see it's everywhere. So that condition really couldn't have been recognized before the advent of those imaging techniques. There are changes in diagnostic criteria that happen over time. There are remarkable differences in medical care and medical intervention, both depending on the place that you are and certainly over time. So, for instance, congenital hypothyroidism untreated is a devastating disease causing short stature, severe intellectual disability, many other features. But hopefully, people with congenital hyperthyroidism are identified very quickly and are treated with, synthetic thyroid hormone, thyroxine, and do very well. And those features really aren't a feature anymore because they don't have that disease anymore. There are cultural considerations, including, who can examine whom in what society. But the biggest problem for us is medical subspecialty bias. So, once people go into a medical specialty, they only focus on that organ system and they really don't pay much attention to the rest of the patient. It's kind of a funny thing. So, for instance, an ophthalmologist, may have all kinds of tools to be able to tell you everything there is to know about the eye. But honestly, if somebody came in wearing pants, they might not notice that the leg is a prosthesis. They just might not notice. They're looking at eyes. So they'll write about eyes and they don't notice the rest of the patient. So we have, on many occasions, seen that, wait, the renal people are writing about this and the ophthalmology people are writing about this and the hematology, that's all one thing, but they don't know because they're looking at their own organ system and they're writing about it. Ascertainment of the phenotype at different stages of development. So a newborn obviously can't do what an adult can do, and the phenotype changes with time. Phenotypic diversity at a locus is the fact that one gene can cause many phenotypes. We talked about that with FBN1 already. Genetic heterogeneity is the fact that some conditions can look exactly the same clinically and be caused by many different genes. An example is cardiomyopathies. Is a hypertrophic cardiomyopathy has, I think I've lost count. It's probably 50, maybe 70 or 100 genes already that cause it. Okay. So how do you deal with that? So OMIM deals with that genetic heterogeneity by lumping and splitting. So the phenotypic series, this is Seckle syndrome. It's a form of extreme dwarfism. You're really, really tiny. And there's a number of genes that cause it. So we break them out and call it Seckle 1 through 9. They're numbered. They're shown in this table based on chromosomal location going from 1 to 22. But they are numbered based on when they're first reported. So this table shows you the location, the name of the disease, its inheritance pattern. This you can read more about, but it just means there's mutations in this gene. That's what 3 means. The phenotype MIM number, the gene for it, gene name, and the MIM number for the name. And these are all, everything in blue is a hot link. Okay, so here they are lined up next to each other. You can see they're actually fairly similar. They're very small, very, very small people. These are phenotypic series. We have many of them. There are currently 584 phenotypic series describing more than 5,130 disorders. Disorder may live in more than one phenotypic series. Okay. Now I'm going to tell you a little bit about what's in OMIM. I am going to, okay. So this menu bar exists on the top of every page in OMIM. It's always there. This will take you to statistics. I want to show you the little contact us button that you can, if you find an error, have a question, want to suggest a paper, this is how to contact us. It's a form that lets you say who you are, which entry you're worried about, and then fill in what kind of question you have. Is it a technical? Is it an error? Is it a suggestion? And then fill that in. It takes a second. I'm going to tell you more about MinMatch. This is where help exists, and help is extremely useful. Lots and lots of FAQs in here in case you run into things you're confused about. Okay. The other thing is there are video tutorials, and I'll tell you more about those in a minute. But they're each short and very useful, and almost everything I'm going to tell you about in an hour is present in 5 minutes in there. Okay, so this is an update as of yesterday of the number of entries in OMIM. So I told you this was a gene. This is a phenotype whose molecular basis is known. There are still 9 entries that are lumped entries. This used to be a much larger number. We'll get rid of these 9 by the end of the year. So there are no more lumped entries. That's just a holdover from many, many, many years ago. A percent sign means that this is a phenotype and its molecular basis is unknown. It might be mapped, it might not be mapped, and this number, as you can imagine, continues to go down as we find more and more disease genes, or that number actually for all of them. And some entries don't have any symbol in front of them. That can be all kinds of things. It can be a thing we weren't sure was Mendelian. It can be a thing we weren't sure was different from something else that somebody described. It can be, that the ability to synthesize vitamin C in humans is a pseudogene. We can't do it. It's all kinds of stuff because, MIM it's a big, it started out as somebody's, collecting interesting information. It's still many people collecting interesting information, although we don't add many curiosities anymore. And this, I wanted to make sure I told you, a MIM number beginning with a 1, 2, or 6 just means it's on an autosome. When it was a catalog of dominant and recessive conditions, a 1 meant dominant and a 2 meant recessive. But we figured out in 1994 that, this wasn't really holding because some had dominant and recessive in the same, associated with the same gene. And so every autosomal entry now has a 6. X-linked is 3, Y-linked is 4, and mitochondrial begins with a 5. So it's always a 6-digit number, but those are the starting numbers. Okay. We are currently we count 5,009 genes causing more than 7,600 phenotypes as of yesterday. And again, I want to reiterate 30% of disease genes cause more than one disorder. And some disease genes were reported last week, right? So they haven't even had a chance to be found with another disorder. So we really are constantly learning. Now, this is a narrower breakdown or a more refined breakdown of the information about the genes and diseases, and they may appear with more than one. So there are 4,600 genes that cause single gene disorders and traits, 6,600 of them. There are almost 500 causing more than 600 susceptibilities to common disease or infection. Non-diseases are things like platelet size, bone mineral density, quantitative trait locus, height, things that are not, absolutely non-diseases. They're traits and they're genetic, but they're not diseases. And then these somatic cell genetic disease, a very few of these are cancers. The vast majority of these are conditions in which you must be mosaic. If every cell in your body had it, you wouldn't have been born. So you have to have some cells without the variant and some cells with in order to manifest. And that's what this number is really, Okay. Now, I told you we don't do all. Here's what we do. We select examples that include the first mutation or few mutations to be discovered, those with a very high population frequency, those with a distinctive phenotype, those with historic significance, those that have a weird way of causing a mutation, and those that cause disease in a strange way, and then distinctive inheritance. So if there are particular variants that are associated with a recessive form of deafness and different variants are associated in the same gene with a dominant form of deafness, some examples of both of those would exist in that entry. Okay. Then we only catalog select variants. I think I've hammered that one home. There are extensive topical links to many variant resources, including population and disease-based. So we link to Nomad, we link to Exact, we link to locus-specific, locus-specific databases, to ClinVar, to many, many different databases where there are many more variants cataloged. And for many of these, the quickest way to get into the right page of that resource is just to go from OMIM to that link, whatever you're looking for. Inheritance resides with the phenotype. Okay, genes just Mendelize. You give one copy or the other copy. They don't care. The inheritance has to do with the allelic number that you need. And so even a condition that only is described as having de novo variants will be classified in OMIM as dominant, because if that person could have a child, and maybe it's got zero fitness and they won't have a child, but if that person could have a child, it would have a 50-50 chance of being passed on. So that is a dominant condition. Okay. Now, on occasion, people write something and they made a mistake. And they find out that what they called disease causing was actually, there was a different gene with different variants that was responsible for the disorder in that person, or there's some other problem. And so then we will put in reclassified variant of unknown significance. And that's what that says. And this explains why. And it says what the papers are. So you can go right to the paper to figure out whether you agree with us or not. Okay. Now, I'm going to run through a few slides that just tell you the kind of links that we are, the actual links. Is no entry will have every link. Again, it's only going to have the topical links, the useful, the ones that actually have a page on that topic in that resource. But we link to Ensemble, MitoMap, NCBI Map Viewer, and the University of California at Santa Cruz UCSC Genome Browser from Genome. We link to different DNA resources, Ensemble, NCBI RefSeq, and UCSC. We link to protein resources, UniProt, the Human Protein Resource Database, and the Human Protein Atlas from Protein. The gene, many links. We go to gene cards, we go to the gene ontology, we go to the nomenclature, we go to pathways, and for the gene info, and KEGG is the Kyoto resource. We go to the gene information in that. Marvel is a page with also links to many resources and nice overviews. Monarch, NCBI gene. This is PharmGKB. It's about pharmacogenomics. We go to that link. That link is only present if there is some information, and UCSC. I told you there are lots of variation links. I wasn't kidding. And many clinical resources, including newborn screening, including what OrphanNet has to say, a gene review if it's there. And I'm going to go through some of the details of some of these in a couple minutes. Many animal model resources, cell lines if you want to do experiments on these, and pathways. So KEGG has pathways, and Reactome also shows pathways. Okay. Now, in this case, I want to, and I think most of this audience won't be searching by features, but it's a very useful way to use. I mean, if you're a clinician and you're looking at somebody and you're trying to say, well, what could this be that I'm looking at? You put in the features of the person you're seeing. And in this case, I've just typed web neck short stature with nothing, just web neck short stature. So I got a whole lot of entries. They will pull to the top the entries that have all of those words. And they will also, you can see, preferably pull to the top the one where it exists in the title. Okay? So, and there's a built-in thesaurus that the only thing I could find to match here was webbed. So it gave me many different words for webbed. Okay? If I had put this in as webbed neck with quotation marks, which is a proximity search, and short stature with quotes, I will only get 62 entries, which will make my life easier because I have to read less. And again, I pulled to the top the one that has that in the title, and then all of these are really the things I was looking for, but you weren't supposed to know that. And here you can see the thesaurus worked much better because it gave me a synonym or for web neck, pterygium coli, and it also gave me many different synonyms for short stature. And you can decide to use them all or none. That's totally up to you. Okay, so in this case, I'm just showing you a little bit more. I typed in dwarfism. It gave me synonyms, but some of them are hard-coded and you don't have to worry about. So things like that gallstones is the same as cholelithiasis, or typing first, or kneecap and patella, or British English and American English spelling. All of those are back-end hard-coded. Don't worry about it. It's not going to come up as, it'll just do the search correctly for you. Okay. Then, okay, I got to go faster. These are some of the features. And now I just want to remind you that there's these two tutorials. And I'm going to spend, I think, a second on MimMatch. And you can create an account which you'll receive an email, and that lets you know about new gene phenotype relationships. That's right there. You can follow specific genes or phenotypes if it's an interest of yours. You can also look at phenotypic series, and you can click that, and you can want to know about a new one, want to follow one, want to know if something's new or added to it. You can select that. You can make a custom list, which is like your table 1 of your review, and you just put in the MIM numbers for either a gene list or a phenotype list, and you get table 1. So here is a gene list, and here's you get the location, cut that off or leave it there. The gene, the MIM number, what the gene name is, the phenotype it causes, etc. And you can do the same thing with a phenotype, the other way around. And then we've recently created a molecular series. It's only available through MimMatch. So if you're interested in this, please sign up for MimMatch. It's free. You just have to, it's just under registration. And it's a tabular list of phenotypes brought together by a shared mechanism, etiology, pathology, currently with 16 series, including 320 members. And these are the list of them. You'll get these slides, but I'm going to skip through these because I want to get to a couple other things I want to show you. But we have non-protein coding morbid genes currently at 19, will be 21 by the end of the week. Repeat mentions. Again, these are things that are hard to find, and it's good to be brought together into these series. And when you bring them together, you learn things. There's 4 different series for the disorders of the epigenetic machinery, because there are writers, erasers, readers, and remodelers. And so we split those apart. A disorder may live in 2 series. This is spastic paraplegia 93, caused by NFU1. It lives in a phenotypic series of many spastic paraplegias, and it also lives in a molecular series because it's a disorder of iron sulfur biogenesis, for which we have a series. Okay, let me go even faster. I'm going to skip this. This just shows you the gene graphic for that. And I'm going to skip. These are regulatory sequences. This is positional variation due to structural variation. So different parts of the genome brought together, creating extremely unusual phenotypes. OMIM sits on an API. The website itself, if you go in through omim.org, you are in its API, but you can do batch searches and large queries from it. It's very robust. People really like it. The help documentation is apparently very useful. I don't use it. I use the front end. But anyway, you can retrieve information in XML, JSON, Python, and Ruby. This is what it looks like in the JSON format. And you can see when you pull it back that it pulls in SNOMED, CT, UMLS, HPO mappings are available through the API if you're doing batch queries. Okay. I'm now going to go into topical links to other resources because I want you to know these resources exist. So I went into Noonan syndrome, and there's 2 things I want you to show. Anytime you use a web resource, look at the date stamp. Absolutely critical, look at the date stamp. So in OMIM, sorry, I lost my mouse. There you go. You'd go to contributors, and you would see that this was last updated some years ago. So the other thing is, if you're a clinician and you want to know what something is and how to manage it, Gene Reviews is incredibly helpful. OMIM has some management, but it's really not the management resource. So this is what a Gene Review is. And if I click on that, I go to that Noonan syndrome. It happens, it was just updated in June, so it couldn't be hotter off the press. There are 925 of these. They're authored by experts. So Amy Roberts is an expert in Noonan syndrome. It follows a standardized format right there. And it's formally updated with new peer review every 4 to 5 years. But if new information is added between that, the staff of Gene Reviews will update the entries. Very, very useful resources. This is to show you 2 other resources. So I went into PTPN11, which is the gene that causes Noonan syndrome 1. And normally it's toggled open to variation. I've toggled open to clinical resources. This is the ClinGen dosage resources and the ClinGen gene validity resource. If I click here, I go to the dosage sensitivity, which says that there's sufficient evidence for haploinsufficiency. So if you're missing a copy of this gene, either because of a deletion or because of a nonsense mutation, you are going to have a disease. That's what that tells me. And this shows me what the gene disease validity group of PTPN of the RASopathy working group of ClinGen thought. So they reviewed this for 4 different diseases. It's definitive for Noonan syndrome, definitive for Noonan syndrome with multiple antigenes, but it's disputed. It does not cause Costello or cardiofaciocutaneous syndrome. Yeah. And in OMIM, we just don't talk about these two because it doesn't cause it. ClinGen specifically reviews it to make sure that, if somebody's written anything about it, they'll say no. It is a resource on clinical relevance of genes and variants. It reviews gene disease validity. It has a variant curation group that looks over particular variants of particular genes, and they're experts in that. The people who are doing that are experts in that gene. The dosage sensitivity I showed you. And there's also a clinical actionability group that looks at the strength of the evidence that allows you to select pathogenic variants in particular genes to be included in secondary findings from exome or genome sequencing. So you use ClinGen to go to look at these things. Okay. And then I want to spend a second on GeneMatcher, which is a resource we built. It sits on top of OMIM data, and it allows you to look, and there's a very good tutorial. I'm only going to spend a second on this, so please read the tutorial or see the tutorial. It's only 5 minutes. It allows you to put in a region, a different genomic region by variant. And it can be because of a deletion, or it can be because of regions of homozygosity. And you actually can compare 2 siblings if you want using this. You have to pick the build, and then you put in the coordinates, and you can either look at what they share, or if this one's affected, this one's unaffected, what they don't share. And this is what it looks like, and this is what you see. And then in this case, we have a retrieval. These are all the RefSeq genes. The next is the OMIM genes overall, and then the OMIM disease genes, and this divides it into dominant and recessive. And then you can pick some of these to just compare, and you'll get the side-by-side clinical synopses from OMIM that day. Tomorrow could be different if we change something. And then in this case, you can also, in this information, it may have links to ClinGen Validity, ClinVar, Nomad, GTR, and the UCSC Genome Browser, depending on what's available and what you asked for. The other thing is that you can lift over to HG38 if you came in HG37, which most of the time they do, and this will give you more results. In this case, I also decided to narrow my search because I wanted to look at fewer things, and I said this patient has microcephaly and seizure. That lowered my number to 39, and I can again see what these are, and I can compare them as we just did. Okay, the GenCC, it's gencc.org. This is the homepage. If you click here for GenCC database, it's a list of 5,553 genes with classifications from different submitters. And it goes from definitive, strong, moderate. Supportive means somebody didn't actually score it this way, but it's in there as real. Currently, that's orphanate. OMIM will be in this spot because we're not doing this level of evaluation. Limited, disputed, refuted, animal model only, or no known human disease relationship. And these are the submitters, many different clinical labs around the world, as well as this Franklin by Genovox is an AI tool, etc. And then in this case, I searched by phenotype. I put in Noonan syndrome, and I got the genes that are implicated or disputed. So A2ML2 does not cause it. It says limited or disputed, and here are the ones that cause it. And when I go in, I picked NRAS to go into more detail, and I can see who said what about NRAS. Okay. And then I open NRAS, and I can see all the details right there and what date they submitted it as well, or the date they submitted and evaluated. Okay. GeneMatcher, you heard a little bit about GeneMatcher. It is a tool. We're at 9, so I can stop if you want me to, or I can keep going for a couple minutes. GeneMatcher is a tool for connecting and identifying novel disease genes, or for ones that are already reported, but you're trying to build your cohort. You can also use GeneMatcher for that. Very, very easy to use. You create an account. You submit a candidate gene of interest, hopefully with variants or the type of variant and the phenotypic features. You will instantly, you hit submit within a minute, you get an email notification of a match or if there isn't one. And you can also connect from GeneMatcher to any of the Matchmaker Exchange nodes. So these are sometimes international, sometimes national, and sometimes project-specific nodes that have exome and genome sequencing information about patients. So you can, rather than entering it or figuring out how to find all these, you can go straight across GeneMatcher and query these other resources. This is the data as of August 1. There's a lot in there. This is the matches, so you can't even see that there's blue and red in here, so I blow it up so people can see it. We continue to grow. There are GeneMatcher investigators in 114 countries. Africa is underrepresented. So if you come from one of these countries and people aren't submitting possible candidate genes for novel disease genes, talk to your colleagues or do it yourself. And this is the number of publications and the number of new genes. There's still a lot of genes, almost 3,500 genes without a match. So we still have more to learn. We always have more to learn. This is the number of cases and unique genes and other resources. And again, this really increases the strength of the ability to connect and make these matches. And that's me. And I'm happy to take any questions now, or you can reach out to me anytime. And I hope this was useful for you. And if it wasn't, my apologies. Okay, I will stop, escape, and I'll stop sharing. Sorry. And you can ask me questions.
Thank you very much, Professor Ada Hamosh. That has been a very enlightening and very detailed presentation. And thank you for all the resources that you have shared, which I believe the scientists, the clinicians, as well as the researchers who are here would be able to use in the future. So we, at this point, would like to invite some questions. We're a little over the 3 p.m. When we're supposed to have closed. So we can invite a few questions before we do that. And as people prepare, let me just read out one question that is in the chat. So one basically asked if the presentation will be shared, and it's been responded to, it will, as well as the slides. From Louise Misa, she's asking, she says, thank you very much for the presentation. And she's asking if when we are looking at all the genes involved in a symptom, what is the approach series that we should use? That is from Lisa.
Yes. So if you are looking, so I would start then with that symptom or feature. But there's two things to be aware of. If the feature or the symptom is extremely common, you're going to get a really big response, right? So just be ready to filter through a lot of things. If you are looking for something very like an unusual constellation, let's say there's a patient with a heart defect and a kidney problem, one kidney and I don't know, a ventricular septal defect. If you're too narrow or you have too many things, you might get nothing because you went too narrow. So if you have heart and kidney, you might do heart and kidney and see how many things come up that have the combination. It's that number will still be large, but you can look through it. But the molecular series is really a very specific thing. It's a small group of things where we've brought together similar mechanistic things. So these triplet repeat expansions or alanine tract expansions, that is about being able to see are there relationships. So for instance, I went through that slide very quickly, but the non-alanine tract expansions, the triplet repeats, are almost all progressive neurological diseases, whereas the alanine tract expansions are quite variable, generally static, and often like a congenital anomaly, not necessarily a progressive neurological disease. So that's very interesting. The one other thing I don't know what kind of conditions you're looking for, but in OMIM, because it's a 60-year-old resource, if you're looking for everything that might have developmental delay, intellectual disability, or mental retardation, nobody uses the words mental retardation anymore, although I like it because it describes something very clearly. It's now pejorative, so people don't want to use it. search with all of those because they may, depending on the timing of that entry, it may or may not have it all. So you have to be a little creative with what you're doing. Any other questions from anyone? I'm happy to answer questions.
Thank you. So that is all that was in the chat. From those that are online, any questions? Feel free to raise your hand or just unmute yourself and ask the question.
If not, we are really over time. So I thank you all for the opportunity to speak to you. And I hope that if you, I'm happy to have this shared broadly. So please feel free to distribute to anyone who'd like it. Thank you.
Thank you so much, Prof. And on behalf of the organizers, we would like to express our sincere gratitude to our speaker and to all the participants. And of course, we're looking forward to the next webinar. And for now, take care and stay well. Bye-bye.
Take care. Bye-bye.
Bye.